Hello friends, today we will learn about animal cell culture, that is general consideration and scale up. So let's begin. In the laboratories, small scale culture of cells in flask, usually 1 to 5 liter volume, are done for establishment of the cell lines. Such cell lines, such cell cultures are useful for studying the morphology, growth, metabolism, etc. Large scale Large scale cultures are required for semi-industrial, that is 100 to 1000 capacity, and large scale industrial, that is 5000 to 20000 capacity. Use of cells for production of wide range of biologically important compounds. Example: enzymes, antibiotics, antibodies, hormones, interferons, plasminogen activator, interleukins. The term fermenter and bioreactor are in common use while dealing with the industrial use of cells. A fermenter usually refers to the containment system for the cultivation of prokaryotic cells, that is bacteria, fungi, which while a bioreactor grows the eukaryotic cells, that means mammalian and insect. The term fermenter and bioreactor are in common use while dealing with the industrial use of cells. A fermenter usually refers to the containment system for the cultivation of prokaryotic cells, bacteria and fungi. Prokaryotic cell means bacteria and fungi, while a bioreactor grows the eukaryotic cells, that means mammalian or insect. Scale-up refers to the process of developing the culture system in stages from a laboratory to the industry. Scale-up, although tedious, labor-intensive and expensive, is required for the production of commercially important products. For a better understanding of scale-up, certain basic and fundamental concepts of cell culture should be clear. Now let's know about cell culture general consideration. There are several parameters that need to be considered for appropriate growth, proliferation and maintenance of the cells in culture. A good understanding of these parameters we will now discuss, that is necessary for scale up. First point is cell quantitation. Second is equipment and medium. Third is pH and buffer systems. Fourth is oxygen. Fifth is growth kinetics. Sixth is types of culture processes, other practical considerations. Now let's know about cell quantitation. The total number of cells in culture can be measured by counting in the hemocytometer. It is, however, not possible to identify the viable and non-viable cells by this method. Now let's know about cell viability. The viability of cells can be detected by use of the dyes. Example: tryphan blue. Tryphan blue. The principle is based on the fact that the dye is permeable to dead cells while the viable cells do not take up dye. Now let's know about indirect measurements for cell viability. The viability of cells can be measured by their metabolic activity. Now let's know about the common parameters. And first is glucose utilization, second is oxygen consumption, third is pyruvate production, fourth is carbon dioxide formation. In recent years, many laboratories have started measuring the activity of lactate dehydrogenase, that means LDH, to detect cell viability. Dead cells release LDH and therefore this enzyme can be used to quantitative measure of the loss of cell viability. Now let's know about equipment and medium. First equipment is culture vessels. The material made up of glass or stainless steel are commonly used for cell culture. Borosilicate glass, example Pyrex, is preferred as it can better withstand autoclaving for suspension cultures, wherein cell attachment to the surface has to be discouraged. The culture vessels are usually treated with silicone or siliconization. Now, second thing we use is medium and nutrients. Appropriate selection of the medium is done based on the nutritional requirements and the purpose for which the cultured cells are required. Eagle's basal medium and minimum essential medium are the most commonly used. The media may be supplemented with serum. Additional feeding of certain nutrients is often required as they are quickly utilized and get exhausted. These include glucose, glutamine, and cysteine. For suspension cultures, media lacking calcium and magnesium are used since their absence minimizes the surface attachments. Now, let's know about non-nutrient medium supplements. Certain non-nutrient compounds are often added to the medium for improvement of cell cultures. Sodium carboxymethyl cellulose addition to medium helps to minimize mechanical damage that may occur due to forced aeration or the forces generated by steered impeller. Polyglycol in the medium reduces foaming in stirred and aerated cultures. Now let's know about pH and buffer systems. The ideal pH for the animal cell culture is around 7.4. The pH below 6.8 inhibits cell growth. The factors that alter pH include the stability of the medium, type of buffer and its buffering capacity, concentration of glucose and headspace. The commonly used buffer of the in vitro culture carbon dioxide bicarbonate system, that is 2 to 5% carbon dioxide with 10 to 25 mm NaOH is comparable to the blood buffer. The presence of phosphate in the medium improves the buffer capacity. Some laboratories use HCPES instead of bicarbonate for more efficient buffering. As glucose is utilized by the cells, pyruvic acid and lactic acid are produced which can alter the pH. If fructose and galactose are used, the acid formation is less, but the cell growth is reduced. Now let's know about oxygen. Oxygen has to be continuously supplied to the medium throughout the life of the culture. This has to be done without causing damage to the cells. Oxygen can be supplied to the cells in one of the ways. Let's know about these ways. First is surface aeration. In closed system static cultures, the head space is used for the supply of oxygen. For instance, in a 1 liter flask with 100 ml medium, 900 ml of the space containing air has about 0.27 gram of oxygen. This oxygen is capable of supporting 10 to the power 8 cells for about 450 hours. Now let's know about spurging. The process of bubbling has through the culture is referred to as a spurging. This is an efficient means of oxygen supply but may often damage the cells due to effects of the bubble on the cell membrane surfaces. Use of higher air bubbles minimizes the damaging effect. Now let's know about membrane diffusion. Adequate diffusion of oxygen into the culture can be obtained through silicon tubbing, which is highly permeable to gases. This approach, however, is inconvenient besides the high cost of silicon dubbing. Now let's know about medium perfusion. The medium is perfused through an oxygenation chamber before it enters the culture system. This method ensures good oxygen saturation. Medium perfusion is in effect used in glass bead system and microcarrier systems. Now let's know about growth kinetics. The standard pattern of growth of cultured cells following a lag phase, an exponential, that means log phase, and a stationary phase occurs. Now let's know about growth of the cells usually means an increase in cell numbers. However, increase in cell mass may occur without replication. There are there are two terms, some terms that are commonly used to represent growth of cultured cells. First is specific growth rate, that means the rate of cell growth per unit amount of biomass. Second is doubling time. Doubling time is the time required for a population of cells to double in number or mass. Third is degree of multiplication, that means the number of times a given inoculum has replicated. Now let's know about types of culture processes. There are different culture processes and growth patterns of cells. We will now discuss. First is batch culture. In this technique, when the cells are inoculated into a fixed volume of the medium, they utilize the nutrients and grow the simultaneously accumulate metabolites. As the nutrients get exhausted, toxic waste products accumulate and the cell multiplication ceases. Further, the cell density drops due to death of the cells. Batch culture is a standard technique. Several modifications have been made to increase the proliferation of cells besides prolonging their life. The other culture processes now we will discuss. First is feed batch culture. There is a gradual addition of fresh medium so that the cell proliferation is much higher than the batch culture. Thus, in the feed batch culture, there is an increase in the volume of culture. Now, let's know about semi-continuous batch culture. A portion of the culture medium is intermittently replaced with an equal volume of fresh medium. The growth pattern of the cells is fluctuating with a rapid increase in cell density after its replacement of the medium. Now let's know about continuous perfusion culture. This is a continuous addition of the medium to the culture and a withdrawal of an equal volume of used cell-free medium. The continuous perfusion process may close or open for circulation of the medium. Now let's know about continuous flow culture. In the continuous flow culture, a homeostatic condition with no change in the cell numbers, nutrients and metabolites is attained. This is made possible by a balance between the addition of the medium and withdrawal of medium along with cells. This is mostly suitable for suspension cultures. Other practical considerations besides, let's know about the other practical considerations. Besides the parameters we discussed till now, there are several other practical considerations for in vitro culture and scale up. Let's know about this. First is culture surface area. The available surface is important for cells to grow. In general, the culture processes are planned in such a way that the surface area is not a limiting factor. Next is inoculation density of cells. As such, there is no set rule for the density of inoculation. However, inoculation with high density is preferred for better growth. Next point is growth phase of cells. Cells in the late exponential phase are most suitable for inoculation. The cells at the stationary phase should be avoided since they have either prolonged lag phase or no growth at all. Now let's know about steering rate of culture. The steering rate of different culture lines are developed in the laboratories. It is usually in the range of 100 to 500 rpm for most of the culture. Temperature of the medium. Let's know about the temperature of the medium. It is advisable to warm the medium to 37 degrees centigrade before adding to the culture. Now let's know about the scale-up. Scale-up involves the development of culture systems in stages from laboratory to industry. The methodology adapted to increase the scale-up a culture depends on the proliferation of cells and is broadly divided into two categories. First is scale-up in suspension. Second is scale-up in monolayer. Now, let's know about scale-up in suspension. The scale-up in suspension is preferred method as it is simpler. Scale-up of suspension culture primarily involves an increase in the volume of the culture. Small scale generally means the culture capacity less than 2 liters volume, and steered suspension cultures it is usually necessary to maintain cell strains in steered suspension cultures by agitation of the medium. The steering of the culture medium is achieved by a magnet encased in a glass pendulum or by a large surface area paddle. The steering is usually done at a speed of 30 to 100 rpm. This is sufficient to prevent sedimentation of cells without creating shear forces that would damage cells. Now let's know about static suspension cultures. Some cells can grow in suspension cultures without steering or agitation of the medium and form monolayer cells. However, static suspension cultures are unsuitable for scale-up. Now let's know about factors in scaling up. For appropriate scale up, the physical and chemical requirements of the cells have to be satisfied. Now let's know about the physical parameters. Configuration of the bioreactors. First is configuration of bioreactor. Second is supply of power. Third is steering of the medium. Now let's know about the chemical parameters. First is medium and nutrients. Second is oxygen. Third is pH and buffer systems. Fourth is removal of waste products. Some of the relevant aspects of the factors in scale-up have already we discussed under cell culture and general considerations. The most commonly used technique for steered suspension culture now we will discuss. First is steered culture. The size of the steered flask is in the range of 2 to 10 liters. It is fitted with a magnetized rotating pendulum and two side arms, one of the addition of cells and medium and the other for the supply of carbon. The steerer culture vessels is autoclave and is then set up and then it is set up. The flask is seeded with the culture. Then medium along with an anti-foam agent is added. The flask is connected to carbon dioxide and steered at a speed of 60 rpm. The flask is incubated for about 2 hours. The contents of the small steerer flask are transferred to a large flask and entire setup is restarted. Incubation at 37 degrees centigrade is carried out for 4 to 7 days. The growth of the cells is monitored daily and the cells are counted. There is a tendency of the cells to enter apoptosis if the concentration exceeds 1 into 10 to the power 6 cells per ml. Now let's know about continuous flow culture. In a continuous flow culture, it is possible to keep the cells at a desired and set concentration and maintain. This is carried out by biostat or chemostat. Now let's know about continuous flow culture in details. Continuous flow culture consists of growing the cells at the mid log phase, removal of the measured volume of cells and replacement by an equal volume of medium. The equipment specially designed for this purpose has the facility for removal of cells and addition of medium. The flow rate of the medium addition can be determined from growth rate of the culture. The medium flow can be regulated by a peristaltic pump. By this technique, it is possible to keep the culture conditions constant rather than to produce large number of cells. The continuous flow cultures are useful for monitoring metabolic changes in relation to cell density. However, these cultures are more susceptible to contamination. Now, let's know about airlift fermenter culture. The major limitation of scaleup in suspension culture is inadequate mixing and gas exchange for small cultures. Steering of the medium is easy, but the problem is with large cultures. The design of fermenter should be such that maximum movement of liquid is achieved with minimum shear to damage the cells. The bubbles a 5% carbon dioxide in air is formed through the bottom of the fermenter. The bubbles formed move to agitation and aerate the culture. These bubbles carry a flow of liquid along with them and release at the top, which goes to the bottom for recycling. It is possible to continuously supply oxygen to the culture in this technique. Airlift fermenter culture technique is suitable for fragile animal as well as plant cells. This fermenter is extensively used in the biotechnology industry for culture capacity up to 20,000 liters. Now let's know about NASA bioreactor. NASA means National Aeronautic Space Administration, USA. Constructed a bioreactor to grow the cells at zero gravity by slowly rotating the chamber. The cells remain stationary and form three-dimensional aggregates, and this enhances product formation. In the NASA bioreactor, there is almost no shear force, hence the cells are not damaged. As the culture chamber stops its rotations, the cell aggregates, sediment and medium can be replaced. Other systems for suspension cultures. Let's know about other systems for suspension culture. First is rotating chambers. The mixing and aeration of the culture medium can be achieved by 2 or 3 rotating chambers. The chambers are so designed that the cell suspension and mixing are high in one chamber while the product and spent medium remain in the other chamber. These chambers are separated by semi-permeable membrane. Now let's know about perfused suspension culture. This also has 2 compartments. The cells are kept in a low volume compartment at high concentration while the medium is perfused in adjacent compartment. The product can be collected in third compartment. Now let's know about scaleup in monolayer. The monolayer culture are anchorage dependent. Therefore, for the scaleup of monolayer culture is necessary to increase the surface area of the substrate in proportion to the number of cells and volume of the medium. As already we discussed, suspension cultures are preferred as they are simple. The advantages and disadvantages of monolayer cultures now we will discuss. First is advantages. First point is change of medium and washing of cells easy. Second advantage is it is easy to perfuse immobilized monolayer cells. The cell product formation, that is pharmaceutically important compounds, example interference antibodies, is much higher. Fourth point is the same set of and apparatus can be repeatedly used with different media and cells. Now let's know about disadvantages. Tedious First point is tedious and costly. Second is require more space. Third is growth of cells cannot be monitored effectively. Fourth is difficult to measure control parameters, oxygen and pH, carbon dioxide, etc. For scale-up of monolayer cultures, a wide range of tissue cultures and systems have been developed. Next is roller bottle culture. A round bottle or tube is rolled around its axis as the medium along with the cells runs around inside of the bottle. As the cells are adhesive, they attach to inner surface of the bottle and grow, forming a monolayer. Now, let's know about the advantages of roller bottle culture. First advantage is the medium is gently and constantly agitated. Second point is the surface area is high for cell growth. Third is collection of the supernatant medium is easy, and there are some disadvantages also. First disadvantage is monitoring of cells is very difficult. Second is investment is rather high. Third point, there is no third point. These are the two disadvantages. And now we will study about multi-surface culture. The most commonly used multi-surface propagator of monolayer is non-clone cell factory. It is composed of rectangular petri dish like units with huge surface area, that is 1000 to 25,000 cm square. The units are interconnected at two adjacent corners by vertical tubes. The medium can flow between the compartment from one end. The cell factory is almost like a conventional petri dish or a flask with multiplier units. The main limitation of cell factory is that it is very difficult to monitor the growth of the cells. The major advantage, however, is its simple operation to produce large number of cells. Now let's know about multi-array disc, spiral and tubes. Spirals and tubes. The surface area for growth monolayer cultures can be increased by using disc, spirals or tubes. They are, however, not in common use as their commercial importance is limited. Now let's know about microcarrier culture. Monolayers can be grown on small spherical carriers or microbeads, that is 80 to 300 micrometer diameter, referred to as microcarriers. The microcarriers are made up of any one of the materials. Now we will discuss. First is plastic, that means acrobeads or bioplast. Second is glass, it may be bioglass or battery glass. Third is gelatin, that may be Ventrigel or Cytotex 3. Fourth is collagen, that may be biospace or biosphere. Fifth is cellulose, that means DE52 by 53. Next point is DEAE dextran, that is cytodox 1 or Dorma cell. The microbead provide maximum surface area for monolayer cultures. This actually depends on the size and density of the beads. The cells can grow well on the smooth surface at the solid liquid interface. However, microcarriers need efficient stirring without grinding the beads. The main advantage with microcarrier culture is that it can be treated as a suspension culture for all practical purposes. Microcarriers can be cultured in stirrer flask or in continuous suspension. In fact, the suppliers of microcarriers provide the technical literature and other relevant information for setting up a microcarrier culture. Now let's know about factors affecting microcarrier culture. Composition and coating of beads, that is gelatin and collagen beads, are preferred as they can be solubilized by proteases. Higher steering speed is usually required. Third is glass beads are used when the microcarriers need to be recycled. Now, let's know about analysis of microcarrier culture. The cell counting techniques are difficult to be used for microcarrier cultures. The growth rate can be detected by analyzing DNA or proteins. Now, let's know about perfused monolayer culture. The growth surface area of the monolayer cultures can be perfused to facilitate medium replacement and improved product formation and recovery. The perfusion can be carried out with pumps, oxygenator, and other controllers. Now let's know about fixed bed reactors. The fixed bed reactors has a bed of glass beads. The medium is perfused upwards through a bed. The cells are grown on the surface of the beads. The products can be collected from the top along with the paint medium. Instead of glass beads, porous ceramic matrix with microchannels can also be used in fixed bed reactors. Now let's know about fluidized bed reactors. In a fluidized bed reactor, the beads are suspended in a steam stream of medium. These beads are porous in nature and are made up of ceramics or a mixture of ceramics mixed with natural products such as collagens. They are of low density and float in the medium. The flow rate of perfused medium is equal to the sedimentation rate of beads. The cells can grow as monolayers on the outer surface and inside of the porous beads. Let's know about the other perfused monolayer cultures. Membrane perfusion, hollow fiber perfusion, matrix perfusion and microencapsulation are among the other techniques for perfusion of monolayer cultures. Now let's know about monitoring of cell growth in scale-up. Monitoring of the progress of cell growth and the culture systems are very important in scale-up. Now, let's know about monitoring of suspension cultures. The progress of suspension cultures can be monitored in situ by measuring glucose, oxygen, carbon dioxide, pH or metabolites produced, lactate ammonia or specialized products found. Example, immunoglobulins by hybridoma cells. The cell proliferation and rate of biomass formation can also be determined by estimating DNA, protein and ATP. Now let's know about monitoring of monolayer cultures. It is rather difficult to monitor monolayer cell cultures for scale-up. This is due to the fact that in most of the technique employed for monolayer cultures, the cells cannot be observed directly to monitor the progress of the culture. In recent years, near magnetic resonance (NMR) nuclear magnetic resonance, that is in short NMR technique is used to assay the content of the culture. The characteristic NMR spectra generated by specific metabolites enables the identification and quantification of metabolites besides detecting the progress of cell growth. Finally finished. Any questions regarding this, you can ask me in the comment box and subscribe my channel to know more updates about biotechnological topics. Thank you.